eastern Nebraska, well watered, I think. Oh, I don't know what this is. Oh, well. Okay. Well, you get the idea. So today I want to talk about phloem loading and phloem unloading, transport within the phloem, and how that gets set up, and I briefly described that last time. But I'll go into more detail today. And then second exam a week from today, next Tuesday, November 30th. So that means Thursday, what do you want to do? I can start with stomata, which is the next thing we're going to talk about. Or we could have that day as a review day, similar to what we did before the last exam.
That one.
That one?
Yeah.
Okay. All right. Okay, that's the plan then. That way I don't have to get all the way through these slides today, right? We can finish up on Thursday morning. There's a fair amount of material to cover, so let's just jump right in. I've got everybody's homework graded too, so remind me, I'll pass that out at the end of class. Everyone did well, by the way, as expected.
Everyone? There wasn't a couple outliers or anything?
You know, really not. I mean, it's yeah, everyone did well. So, okay. Yes. Okay. Transport and flow. Has anybody taken art history? You know, that's a class you didn't want to schedule right after lunch because you go in there and when I was in college, they show slides and you look at slides of paintings and things in a dark room and, you know, kind of nod on. Anyway, this is a famous painting by Norwegian artist Edvard Munch. But what does it have to do with transport and the flow? In 1930, a German physiologist, Ernst Munch, not this guy, Edvard Munch, that's the famous painter, but a different guy with a similar name, came up with a theory of flow and transport that he called pressure flow theory. And it was that circulatory thing that I explained last time. And so that's what I'll go into today. It's still the most widely accepted theory for transport and the flow. The circulation between the phloem and the xylem, circulation of water between phloem and the xylem, driving forces are pressure gradients within the xylem and within the phloem, but those are set up by changes in osmotic potential at one end of the phloem and the other end of the phloem. And that's what I want to talk about today. So the end result is that sucrose or photosynthates get transported from areas of high concentration where they're produced in leaves, let's say, to areas of low concentration in roots and meristems and non-photosynthetically active tissue. So there's a transport of sucrose or other carbohydrates from high concentration to low concentration, but diffusion isn't the primary or even a major factor in transporting that sucrose. It's pressure-driven mass flow, convective flow within the phloem. And the fluid is moving in response to differences in pressure potential. So here's an older reference to explain. This is the famous painting. You know this one? This is really famous. Yeah. Okay. All right, so what do we have? All right, let me explain this thing. This isn't my own picture, this is somebody else's. But let's assume we have a beaker over here and another one over here. And these two beakers are filled with pure water. Okay? And what's more, they're connected by a tube on the bottom that goes through the screen here. pure water. Pure water, so the total potential is 0. Equals the osmotic potential equals the pressure potential, are all 0. Okay. You missed the video.
I have it.
But there's no sound on this system, so we couldn't hear it. We just saw it.
That's
It's like,
terrible.
there's no speaker over here. Oh well. Or else I don't know how to turn it on. Okay, so let's say pure water here, pure water over here. Pure water. We call this one B and this one A. Now, if we suspend a bulb in each beaker, like this, that has a perfectly semi-permeable membrane around it. That means that the reflection coefficient is 1 for all the solutes. So we suspend that in here, and a similar one over here in this beaker, and we connect them with the tube. Perfectly semi-permeable. And now fill this bulb and the tube also with pure water. Pure water everywhere. Water, water everywhere. Okay? And this also has perfectly semi-permeable membrane. Okay, so what happens? Nothing much. Nothing happens. The water evaporates and nothing's going on. No, so interesting. All right, now, let's say down to 2 of these now. Let's say all of a sudden, okay, pure water here, pure water here. So at the beginning, the total water potential inside here is also 0. And that's the pressure and the osmotic potential as well. It's pure water everywhere. So there's no net movement of water into or out of either of these bulbs on either end. There's no movement between either of these tubes or within either of them. But now let's automatically start adding solutes into here. Okay, so let's assume that you snap your fingers, whatever, and solutes appear in there. Something like sucrose. Now what happens? Now we have an osmotic potential here lower than 0, less than 0, negative. The total water potential is also less than 0 now, inside this bulb. It's 0 here, it's less than 0 here. Which way does the water move? It goes in, right? It moves in. A different color. In response to that difference in the total potential, because it's a membrane and it's the total potential, the difference in the total potential is driving the water movement. So water moves in here, or starts moving in here and starts swelling this up if it can, or increasing its pressure potential. And eventually the water moves in until the pressure potential equals the negative of the osmotic potential, and the total potential is 0, and then the water quits moving. But in the meantime, since water is fairly incompressible, this increase in pressure potential here also causes an increase in pressure potential here. So here, when we add solutes, the pressure potential is greater than 0. Initially, the osmotic potential is still 0. There's nothing dissolved in this. But the total potential is greater than 0. Out here, the total potential is 0. So which way does the water move over on this end? It moves across the membrane like this, out into this beaker. So we have water moving in this end, moving out this end, and we have a flow of water through this tube this way. Right? That movement of water from here over to here raises this level a little bit. This level's lower a little bit. That causes differences in pressure that make the water go this way through this tube that connects the two on the bottom. Right? So we keep adding solutes here. Eventually, some of those make their way over here when enough water moves, and they begin to accumulate here. This gets negative, but if we magically remove them, these solutes, when they're in this, when they get over here, we take them out of the system, we can keep this circulation going as long as we continuously add solutes here and continuously remove them over here. We can keep this circulation system going. And this is how things move in the xylem and the flow. In this schematic, well, I can work my way through these slides. In this schematic, this addition of solutes here is called phloem loading. Yeah, phloem loading. Sticking solutes into the phloem. This removal of solutes is phloem unloading. And it only seems like magic. I don't think there's much involved. But there's a lot more that's known about phloem loading than phloem unloading, as we'll get to by Thursday.
Was this actually done? Like
What? This
in experimentation?
I don't know.
Or is it just like a theoretical schematic?
As far as I know, I've never done it. I think it's a theoretical schematic, but there's no reason you couldn't do it. You just need to find a good memory.
Yeah.
That may be the tough part.
I guess I'm just trying to figure out mentally my own theoretical experiment is how you would get solutes inside it. Like,
Yeah. That's
would
it.
just be like.
Well, that's why I say snap your fingers or wiggle your nose or somehow magically get them in there.
Yeah.
But I'm going to talk about that. I'll talk about that more. That's phloem loading, and there's actually quite a bit that's known about phloem loading. Because ultimately, phloem loading involves taking that. These solutes are recent photoassimilates. These are carbohydrates that have been produced from photosynthesis.
Right.
And that process, that production of those photoassimilates happens in the mesophyll cells, in the cytoplasm of mesophyll cells. Somehow, those carbohydrates have to get from that mesophyll cell into the phloem. So how does that happen? Magic. How does that happen? That's the trick. And it's not really magic, but it's complicated. And different plants do it different ways. So it's that. Phloem unloading, who knows? That's black magic as far as we know. I mean, there's very little known about phloem unloading, but it happens too. I mean, it's got to. But okay. Okay. Well, this is Monk's theory, pressure flow theory, right? It explains most of the experimental evidence that exists regarding transport in the phloem, and I'll talk about some of that in a minute. It doesn't explain everything, so there are observations that are in conflict with this theory, and I'll point those out.
So in this experiment, the phloem would be the bottom one?
The phloem would be this one, and the xylem would be this one.
Because it carries the solids.
Yeah, this is carrying the solids and phloem. Sure.
What about kind of thinking in a corn plant or soybean plants
Yeah.
where you have sugars
Uh-huh.
going out to the grain? going
Yes.
up.
Yes, yeah,
That
yeah.
should be silent, right? How-
That's the phloem. That's also the phloem, yeah, going up, sure, that's phloem. Yeah,
It's not going down the flowing?
the transport in the phloem goes whatever direction it goes based on the differences in the pressure in the
Okay.
system. So if you have smaller pressure in the grain, in the phloem near the grain, than in the leaf below, like flag leaf below the grain, then it will move up.
Okay.
Yeah, it's
It's a
up or
still-
down, doesn't really matter, sideways, any direction. It's
Okay.
just the gradients in pressure. Make
Okay,
it move
it's
wherever.
still flowing.
Yeah, yeah, still flowing. Right, right, that's a good question, because you can also have a situation where if you measured water potential, you could have a total water potential in the flag leaf that is more negative than the total water potential in the grain. And yet the solutes are still moving towards the grain. Why is that? Because the total potential isn't driving that movement, it's the pressure potential that counts. Yeah. Okay, but it is a theory, and there are things that it doesn't explain, as we'll see in a minute. Okay, so in this scheme then, this situation here at A, it represents the source. The source tissue, the source, whereas this one over here is the sink, right? This is where photoassimilates are produced. This is where they're utilized. The grain, developing grain, any meristems that aren't photosynthetically competent yet, a stem or a leaf meristem, storage tissues like a carrot or a potato or something like that, roots, anything in the root system that is not photosynthesizing. Those are all examples of sink tissues. The source is typically leaves and mesophyll cells in the leaves where photosynthesis is happening. Okay. Yeah, I think I said all this stuff. Right? Okay, so when you go through these slides later, hopefully you'll be reminded of some of these things. Right? You'll all go through the slides later, right? I mean, it's up to you. Do what you want. But exam a week from today. Okay, here's a different picture. This is from the Plant Phys and Development book, this one, which again I highly recommend if you would like to learn more plant physiology. All right, so in here, this is the xylem, this is the phloem, and they're shown apart, but really they're next to each other. They're very, very close to each other. All right, what's going on? There's a typo. This number should have a negative sign in front of it. All right, but what's going on? Let's look, because not only are the arrows on here with the water moving around, but it's got numbers. So that's always fun. Okay. Let's start here in the xylem. Let's say near the soil, maybe down low in the xylem. Total water potential -6 megapascals. That's made up of a negative pressure potential, a tension in the xylem of -0.5, and a small osmotic component. Okay. Up here, further in the plant, the total water potential may be -0.8 megapascals. That's comprised of -0.7 pressure potential and the same solvent potential, same osmotic potential. So here we have a gradient in pressure from -0.5 to -0.7. Since it acts like an open tube, the water moves this direction in the xylem. The water's moving up here. Now, if we focus on this area where the xylem and phloem are together, here we have a total water potential of -0.8. But in the phloem in that region, the total water potential may be -1.1 megapascals. That's made up of a positive pressure potential and a very negative relative to the xylem. Osmotic potential. Lots of stuff dissolved in the water. So, since this movement from here to here involves crossing a membrane right here outside the sieve element, water is going to move based on the difference in the total water potential. So here we have minus 0.8 megapascals, here we have minus 1.1 megapascals, so the water moves from the xylem into the flow in this region. If you look through here, and if you read this explanation, or actually some of this comes from Park Nobel's book, Physico-Chemical and Environmental Plant Physiology, he calculates a gradient in pressure potential based on the size of the sieve tubes and the dimensions of these sieve plates and things like that, and comes up with a gradient that corresponds with this. So the pressure potential here may be 0.3 megapascal. The osmotic potential minus 0.7, there's a negative sign missing from this number, minus 0.7. And that makes a total water potential of minus 0.4 megapascal. Again, the communication between the phloem and the xylem involves water moving between the two, involves crossing a membrane. So it's this difference in total potential that drives things. And here we have minus 0.4 megapascals, minus 0.6, so water's going to move from the phloem into the xylem down on this end. Within the phloem, plus 0.6 megapascals, plus 0.3, that pressure gradient causes the water to move this way, the fluid to move this way. So this scheme all fits together, and I mean, you can make sense of it in terms of the components of water potential in all these places. What it involves, though, is making a quite negative osmotic potential here and a less negative osmotic potential here by moving solutes into the phloem on this end and removing them on that. Right? So again, that's from this Plant Phys book. You know, let me just check. This picture is from the 3rd edition. You can see this is the 6th edition. I wonder if they'll change that negative sign. They changed the chapter number. Well, it doesn't matter. You can look it up. Right? Here we go. 285. Oh. All right. There's lots of nice pictures in here too. Yes. Yeah, there's a negative sign on there. Okay, anyway, that's how it works. So, what evidence do we have that this theory is consistent with? Well, if you look, positive pressure potentials in the flow, greater than 0 in the flow. If you play around with aphids and let them feed on the phloem, cut their body off and measure the pressure from the end of their stylet, positive pressures in the phloem. So pressure potential greater than 0 within the phloem. Here's an article that talks about it. This graph is from, this table is from their article. They measured this with a sensitive pressure gauge manometer, measured these directly. They also calculated them with measurements of total water potential and then estimates of the osmotic potential based on how much sugar is in there and stuff like that. And you know, they're not exact, but positive. So positive pressures in the phloem that Monk's theory predicts have been observed. high concentration of sugars in the phloem. All right, so here's a table from this reference that shows in cucurbits, high concentrate, these are millimolars, 60 millimolar concentration of sucrose. Well, you can see all these numbers. Raffinose, which is just a bigger sucrose with 6 more carbons added on. Stachyose, add 6 more on. Verbascose, add 6 more. So higher sugars, but high concentrations of all of these sugars in the phloem of these cucurbit plants. So that's consistent with Monk's theory. Right? After all, here's high concentration of something. So that's consistent with Monk's theory. Here's another one. Here's a nice one. This one, I like this one. So here's cowpea, Vigna unguiculata, right? And here's a cowpea that's infested with the virus, cowpea mosaic virus. Cowpea mosaic virus moves in the phloem of the plant. So you can take a cowpea leaf, a healthy one, and rub it with carborundum or something and put some viruses on there and mush them in there so they get into the phloem. If you keep the plant in the dark, the virus stays where you put it. You don't see it anywhere else in the plant. But if you take that infested leaf, and take the plant with the infested leaf, put it in the light so it photosynthesizes, then the virus shows up all over the plant. It's known to travel in the phloem. How do you explain that? Monk's theory explains this. If the solutes that are being loaded into the phloem come from recent photosynthesis, this is the behavior you would expect. And other viruses are similar. Do experiments with them too. So, pretty good evidence. It's all circumstantial, right? But, Monk's theory explains it. Especially this one. But, here's the caveat, right? But, some observations Monk's theory doesn't explain. First of which is different substances can move at different speeds within the phloem. So sucrose moves this fast, something else moves half as fast or twice as fast. That's not consistent with mass flow. Everything should move together at the same speed through the phloem if pressure is driving. Now, it's worth noting this phenomenon has never been observed within a single sieve tube. It's always an array of tubes that can show different things moving at different speeds. So maybe, make up your own idea for how that happens. Maybe things leak out and have to be stuffed back in all the time. I don't know, something. But, okay, so that one may or may not be problematic, but this one really is. Anoxia, lack of oxygen, or cool temperatures reduce the rate of phloem transport. So if you have erase this. We don't need this anymore, right? If you have a leaf, let's say, you have a nice leaf here with a super long petiole. And this leaf is photosynthesizing, and things are moving in the phloem through the petiole out to wherever they're moving. You can measure the rate of this transport by using a stable isotope like carbon-11 or something, or a radioactive isotope, carbon-14. And you can measure a rate of movement in the phloem. If you take this bit of stem, this petiole, and put it in a space and remove the oxygen, or cool it off, you'll affect this rate of transport. What's more, you remember r squared over 8 beta times the gradient. This is what's driving the movement. And you say, this viscosity. Well, when it gets cold, the fluid is more viscous, and it's that interrupts the flow. But this change, first of all, that doesn't explain why no oxygen affects it. But this changes way more than you can explain by changes in viscosity of the fluid with temperature. So how does that work? There's nothing in Monk's theory that says the phloem has to be alive. Those phloem cells have to be alive. There's no need for that, according to what we've just talked about. So how does that work? You know that the cells in the phloem are living, and we know that they're respiring just like all living cells, and that respiration rate is a strong function of temperature. So that and it requires oxygen. So there's something about the transport system in the phloem that requires that seems to require those cells to be alive and respiring to make things work. And Monk's theory, silent on that, doesn't say anything about it. All right, these 1980s papers talk about that. It turns out it's not so easy to do experiments like this, because if you use a radioactive tracer like carbon-14 and feed a leaf, we did this way back in Jerry Mirando's course. You can take you can make carbon-14 O2, right? 14 CO2, 14 CO2, and this is radioactive, so you can use a Geiger counter or something to detect it. And if you feed the leaf 14 CO2, you can watch it as it moves through the plant, by where it's decaying in the plant. Carbon-14 has a really long half-life though. And so when you do these experiments with C14, pretty soon the whole plant is covered with C14 and you can't tell where anything's going anymore, because it's all radioactive, right? Then you have to dispose of this stuff too, which is very expensive. So people don't tend to use C14 anymore very much. But what you can use is C11. That's a stable isotope. Not a stable isotope. It's an isotope with a very short half-life, 20 minutes. C14 has a really long half-life. I don't know what it is. Years and years and years. But C11 has a 20-minute half-life. So you can feed this leaf 11 CO2 and then watch where it goes and change things and watch how things move and change. And it's decaying and turning into whatever it turns into, I don't know, some other isotope of carbon, right? So it's much more amenable to these types of experiments. The problem is 11 CO2 has half-life of 20 minutes. Where do you get it? You order it from Sigma? They're not going to deliver it in 20 minutes. Maybe Amazon with their drones, someday you can get out 20 minutes. I need some C11. But right now, you have to make that isotope right next to where you're growing your plants to do the experiment. And that's why a lot of this work was done at Duke University, where the physics department cyclotron was right next to the phytotron where they're growing the plants. So they run a tube from one to the other, they make the stuff, and they use it for their experiments. And they actually published a bunch of papers in the mid-80s on this subject. So anyway, that's a bit of an aside. All right, but regardless of these observations that aren't explained well by Monk's theory, it explains almost everything else, and so it's the commonly accepted theory of flow transport. So that's it. Okay. Let me talk about flow and loading. Now, let's examine this in more detail. Well, you know that the sieve elements are alive, but they have no nucleus and all this other stuff. And they're associated with a companion cell that comes from the same mother cell, the sieve element and the companion cell together. They're always together. To understand phloem loading, we have to focus on these companion cells. They play a critical role. There's a couple of references. All right, just for review, you remember photosynthesis. This isn't, I don't like this picture very much, but this is supposed to be a chloroplast, this yellow thing. Chloroplast with thylakoid membranes, these green parts are the thylakoid, right? So this is called the stroma of the chloroplast, and the cytoplasm of the photosynthetic cell is all around it up here, right? So what's photosynthesis? 3 parts, remember? The light reactions, that's where pigments in the thylakoids absorb light. An electron, by absorbing a photon, an electron gets bumped into a higher energy level. And then that electron, in a series of reactions, finds its way to a special reaction center chlorophyll, what they call reaction center chlorophyll. And then that reaction center chlorophyll donates the electron to, in photosystem, photosystem 2, to a receptor called Q. And photosystem 1, something they call X, right? And this goes to NADP plus, like NADPH. All right, so you've got an electron transfer reaction here and here. This requires a photon, or a few photons actually, as this did. So this happening in the thylakoid membrane, actually across the thylakoid membrane, these are called the light reactions of photosynthesis, the electron transport reactions. And the end result is production of NADPH, which is a powerful reductant, and it results in an electrochemical gradient, a gradient across the thylakoid membrane. So between the inside of that thylakoid space and the stroma, a gradient in pH and electrical potential. The energy that's found in those gradients across the thylakoid membrane then is used to let protons move back the other direction and synthesize ATP. So the ultimate end products of the light reactions of photosynthesis are NADPH and ATP. Then those things are found in the stroma of the chloroplast, or ultimately wind up in the stroma of the chloroplast, where the Calvin cycle, the second big part of photosynthesis, takes place. This, remember, is RUBP, which is a 5-carbon compound, plus CO2, obviously 1 carbon, make 2 phosphoglyceric acid, 3 carbons, and this step is catalyzed by rubisco, enzyme rubisco. And then of those, you have a complicated pathway that regenerates this 5-carbon RUBP, and for every 3 times around this Calvin cycle, you can pull off a 3-carbon carbohydrate. That 3-carbon thing is either incorporated into starch within the chloroplast, or it's transported out of the double membrane surrounding the chloroplast in exchange for an inorganic phosphate. It's a, what do they call it, co-transporter. If there's no phosphate out here, you can't get the carbon out of the chloroplast. All right. Well, anyway, Calvin cycle, second big part of photosynthesis. Third big part is this 3-carbon compound that you've exported goes through a whole series of reactions and ultimately winds up in sucrose in the cytoplasm of the mesophyll cell, or bundle sheath cell, wherever this is happening, in the cytoplasm. That sucrose now, phloem loaded, how do you get that sucrose from here into the phloem? And as I said, to answer this question, you have to take a look at companion cells. So there are 3 kinds of companion cells that have been classified, I guess. All of them have lots of mitochondria. All of them have many, many plasmodesmatal connections between the companion cell and the sieve element that they're affiliated with. They may or may not have plasmodesmatal connections with other cells, but with the sieve element, lots of plasmodesmatal connections. So, implying that there's a lot of transport of substances between those 2 cell types, the companion cell and the sieve element. All right, the 3 kinds of companion cells that have been identified: ordinary, transfer cells, intermediary cells, differ from each other in at least these following ways. The ordinary companion cell, few plasmodesmatal connections to any other cell besides that sieve element. So lots of plasmodesmatal there, few or none with any other cell around. The transfer cell, also few plasmodesmatal connections with any other cell, but if you look in the transfer cell on the side away from the sieve element, there's a lot of ingrowths of the cell wall and cell membrane, which really increases the area of that part of the cell. That's called a transfer cell. Increased area for uptake from the apoplast. Okay, so here we go. Intermediary cell, they've got plasmodesmata with everything. They make connections with all the cells around. Okay, so these are the 3 main types. And these types have implications for the pathway of phloem loading. Here's coleus. Okay, so some plants like coleus have more than one type. So remember coleus, that's this. Here's one example. Okay, so here's a more recent paper that's looking at transfer cells, one of these types of intermediary cells or phloem parenchymal cells. And we'll see images on that slide. But what I want to say is these people use confocal microscopy. And that's a technique, a microscopic technique, which you can do in the Beetle Center. They have a microscopy facility, and you can go down there and pay them money and do whatever you want with it. Well, they'll do it for you, but you get the idea, right? But it makes a 3D, you get like a 3D image of what you're looking at. Really cool. Anyway, here's some of these 3D pictures. Okay, so this is in Arabidopsis. So here's a minor, what they call a minor vein. So little tiny vein in the leaf, right? So this, they peel off the epidermis, and this part in the box is this part up here. Right? So you can see companion cell. What's the single aspect? Transfer cell. Double esters. Oh, another transfer cell, companion cell, and then here's a bundle sheath cell. Okay, so that's all these things right in here. If you take this YZ axis here and look at it this way, that's what you see here. So here's a bundle sheath cell. Here's a sieve element with a transfer cell next to it, another transfer cell, and you can see these ingrowths of the cell wall that show up here, they show up here, and if you look along this axis, you see this, And here's companion cell, here's sieve elements with a lot of these wall ingrowths right next to them. So this is the type of companion cell here that few plasmodesmatal connections to other cell, but the inner surface of the cell wall opposite the sieve element has a lot of these ingrowths that increase the communication with the apoplasm. All right. Here's some more pictures. Here's a sieve member, here's a companion cell, here's plasmodesmata that connect these two, and you can see some of these wall ingrowths on the cell wall opposite the sieve element. I know some of these things. This one's not confocal, so you confined to one plane. Here's another one. This is, I think it's a plant. Intermediary cells. An ordinary companion cell. These are the sieve elements. Here's the xylem. Here are the bundle sheath cells. Here are the chloroplasts that are doing the photosynthesis, right in place. Ordinary companion cell, intermediary cells. Okay, and yeah, the intermediary cells are the ones with lots of plasmodesmal connections. Yeah. So if we look at these, this intermediary cell, you can see them here. These darker lines, lots of connections here, lots of connections here, some connections here between the sieve element. Connections all over the place with these indicators. So, and here's some other pictures. Okay, so let's get down to brass tacks. In plants that have ordinary companion cells or transfer cells, there's an apoplastic step in the phloem loading process. And the way it works is this: sucrose is produced in the cytoplasm of the mesophyll cell. It's transported from the cytoplasm into the apoplast of that cell, across the plasma membrane. So remember, for sucrose, plasma membrane has reflection coefficient close to 1. It's not going to move there passively. That's an active process. That takes energy for this cell to move that sucrose from inside to outside. Once the sucrose is outside this cell, it tends to have a higher concentration here than over here close to the sieve tube, let's say this transfer cell, because the transfer cell is going to take that sucrose and transfer it into itself, again expending energy. So we have a high concentration of sucrose here, lower concentration here, and the transport's by diffusion, high concentration to low concentration. So the sucrose gets actively exported from this cell, diffuses over to this transfer cell or the ordinary companion cell. This cell then takes that sucrose and transports it across the membrane actively by using energy in the form of ATP, and the sucrose ends up inside this cell. What ends up happening is you have a high concentration of sucrose here, higher than in the sieve tube, and the plasmodesmatal connections, this big giant one here, provide a pathway for this sucrose to diffuse from the companion cell into the sieve tube. This is the phloem loading in plants or in regions of plants that have ordinary companion cells or transfer cells. Okay, so as I said, plasmolemmas, plasma membrane, have reflections coefficient near 1 for sucrose. So the plant has to expend energy to move that sucrose. And what is the energy? The energy is in the form of ATP. So there are ATPases enzymes embedded in the plasma membrane that take ATP and break it down to ADP plus an inorganic phosphate, releases a lot of energy. That energy is used to pump a proton from one side of the membrane to the other. That results in a pH gradient and an electrical potential gradient across that membrane. And the energy that's inherent in that is used by a symporter that will take a proton and a sucrose and let them both back in at the same time. So ATPases provide the energy, and these other porters embedded in the membrane, the symporter, lets the sucrose in with a proton. ATPases are found in the plasma membranes of these companion cells, transverse cells, ordinary companion cells. They're found in the plasma lemma. Interestingly enough, developmentally, these ATPases first appear when the leaf starts to become a net exporter of photosynthate. That's when you see them showing up in the membrane. You can measure these electrical and pH gradients across the membrane, blah, blah, blah. And you know, there's been a lot of work done with these symporters too, and they have names and what do you call it? Structures been figured out and stuff like that. So this is all consistent with the idea of an apoplastic step for phloem loading in plants that use ordinary companion cells or transfer cells. And that's why too, you know, if there is the apoplastic step, you know, that's why these ingrowths in the cell wall provide a lot more space for that kind of activity to happen on, because the membrane, you know, it's more membrane there. So it's consistent with the anatomy too. Evidence for apoplastic pathway. If you look at the apoplast, you can find sugars in the apoplast of photosynthetic leaves. How do they look then? Okay. If you take a leaf or leaf disc and float it in a dish of sucrose, of sugar water, what you'll find is that the minor veins in that leaf disc end up accumulating sucrose. You know, even in the dark or something. Well, there's something that's moving it there. And this molecule, this weird thing here, PCMBS, this is a chemical that inhibits sucrose transport across membranes, but it can't move across the membrane itself. It's a weird one, right? So it acts to block this thing off, right? But it can't, mesophyll side, vein side. It can't penetrate the cell, but it makes this stop working. If you apply that with your exogenous sucrose, you don't see the accumulation of sucrose in the minor veins anymore. Also, this chemical, if you put it on a intact leaf, it blocks phloem transport, or it reduces phloem transport. Not block, but reduces it. So here's the results from experiment showing this. The green dots are the translocation rate of labeled photosynthate, whatever that is, from a sugar beet leaf. Okay, so C3 plant, sugar beet. So here's the translocation rate out of the leaf through the phloem, whatever this number is. And the purple is the photosynthetic rate of that leaf that's doing the transporting. So that's like this. So things are going on, and then you put this chemical that blocks that sucrose transport. You put it on, and the translocation rate immediately drops. But the photosynthetic rate is unaffected. So you're still producing as much photosynthate, but somehow it's not getting out of the leaf because it's blocking that sucrose transporter in the plasma. So this is pretty good evidence of what's going on in those plants, those portions of the plant that have ordinary companion cells or transfer cells. In contrast to that, there are some plants that have intermediary cells. Now these are the ones that have plasmodesmatal connections to all the cells all around it. And in particular, well, Coleus has some, but in particular, the cucurbitaceae, plants in that family, the cucurbits, melons and pumpkins and squash and that stuff, they have intermediary cells, and they work differently. In these plants, phloem loading occurs totally symplastically. There's no apoplastic step. And how does it work? Sucrose synthesized in the cytoplasm of mesophyll cells. That sucrose diffuses from this cell to an intermediary cell through plasmodesmatal connections. The sucrose concentration in this cell is kept low because once it gets here, this sucrose is combined with galactose to make raffinose. So low concentration of sucrose here, high concentration here, means the movement is this way. Raffinose, high concentration here, lower concentration in the sieve tube. It also diffuses through plasmodesmatal connections this way. Some plants don't stop at raffinose, but that's sucrose plus galactose is 6 carbons, so that makes 18 carbon sugar, raffinose. Some plants stick another galactose on to make a 24 carbon stachyose, or a 30 carbon verbascose, and a similar thing happens. Now, okay, that works great. You have to wave your hands a little bit.
Without personifying the plant makes raffinose because it can't diffuse backwards, right?
Yeah, that's what I was just getting to. So that's why the hand waving part is these plasmodesmata are different sizes, and the big molecule can't go back to where the sucrose came from. Sucrose is small enough to make it this way, but raffinose is too big to go back that way. That's what they say anyway.
So it's made in the intermediary cell, basically?
It's made in the intermediary cell, yeah. It's synthesized here. Yeah, from galactose and sucrose, yeah. And then it's too big to fit back through that little hole where it came from. Or, if you glom some more things onto that, same thing. So that's why these intermediary cells have plasmodesmatal connections to all the cells around them, because there's stuff moving through there. And this is called the polymer trapping model. Okay. All right. Okay. So you can see, if you look at what is going on, higher concentration of sucrose has been observed here than in intermediary cells. And the enzymes for the synthesis of raffinose and stachyose and everything are found in these cells, consistent, and different sizes of plasmodesmata in different places in the system. So there is experimental evidence for this idea too, and it's not just somebody made it up late one night. Okay, so this is polymer trapping model, totally symplastic movement versus the apoplastic movement that I talked about with ordinary companion cells and transfer cells. Okay, so plants here's another plant that uses this polymer trapping model. I don't know this one, Technical Maria capensis capensis. But they also have some interesting anatomy that goes along with these. So here's a bundle sheath cell. These are bundle sheath cells with their chloroplast, intermediary cells, and here are the sieve elements. And somehow stuff comes from here into here, there, through plasma-dismal connection. Okay, that's phloem loading. Phloem unloading, look, only one picture. Phloem unloading, not a lot known about it, but happens in reverse. The good thing is, here, sucrose, let's say sucrose is in the phloem, sucrose is moving from area of high concentration to lower concentration, say in developing seed. That sucrose moves from the phloem into the seed, gets converted into starch, let's say. That keeps the sucrose concentration low in the seed, high in the phloem, and just diffusion will make it move from the phloem into that cell, if there are plasma-desmotic connections in them. So that's good. Also, one of the big questions is symplastic movement or apoplastic movement. Invertase is present in the apoplast of some plant things. And invertase takes sucrose and breaks it down into fructose and glucose. So the presence of that enzyme suggests maybe something's going on there. In many sink tissues, there are lots of plasma-desmotic connections between the sieve element companion cell complex and the parenchymal cells around it, and other cells around it. So those plasma-desmotic connections would provide a pathway for diffusion of sucrose. But in some tissues, for example in seeds, the maternal tissue and the filial tissue, completely separate. No plasma-desmotic connections at all between those two tissues in any plant that has seeds that develop. Totally cut off plasmodesmotically, if I could say that word. That means there must be an apoplastic step somehow, right, in that tissue. Okay, well, you can read this. It may be, and it probably is true, that this phloem unloading process varies with different tissues and different plants and things, just like phloem loading does. But really not much is known about it. In this 1990 paper, I like it because of the title. What is phloem unloading, right? So it's kind of like today, right? Solar-induced fluorescence. What is solar-induced fluorescence? We can argue about that some other time. Okay, but that's phloem unloading. So there you have it. Phloem loading, phloem unloading, Monk's theory of transport, and please come on Thursday with questions for me. And otherwise, I'll try to review as what I think is important, and we'll have at it. All right? I've got your homework.